Greetings everybody who's watching this video, and I am Snaula from University of Agriculture Festabad Subcampus Budebala Plant Breeding and Genetics Department. I am very pleased to be the part of this conference, and this is a preview of our presentation. The title of our poster is Prebreeding, Bridging Untapped Genetic Resources with Breeding Programs. First of all, here comes abstract. As we all know, world nowadays is surrounded by a lot of stresses. In the same way, agriculture is also facing the many challenges, and one of the leading causes for this is the narrow genetic basis of crops, and there is a need to broaden these narrow genetic bases. Basically, pre-breeding is the best way for this and for broadening our genetic basis and developing new cultivars. And here is the introduction of pre-breeding. Pre-breeding basically refers to all the activities which are performed for the identification of desired traits or gene of interest from germplasm, wild resources, etc. It involves the activities from collection of germplasm to the development of pre-breeding population, which is further used by the breeders to develop new cultivars. Pre-breeding is used for the improvement of our crops, for improvement of our cultivars, like in the sense it can broaden genetic diversity, and it introduces new genes in the population, as well alleles also. And in this way, first step is a collection of germplasm, and germplasm is collected from different sources like land races, wild species, wild relatives, current cultivars, and there are many classes like for germplasm, which are base collection, active collection, and breeders collection. Breeders collection is also known as working collection. Here is the base collection first. Base collection includes accessions which are stored for long duration, and it can be preserved for more than 50 years, and it is provided with temperature which varies from minus 10 degrees centigrade to minus 20 degrees centigrade, or sometimes minus 150 degrees centigrade to minus 195 degrees centigrade with low relative humidity. Active collection and breeders collection. Active collection includes the germplasm which is actively distributed to the scientist with 0 degree centigrade temperature and 8% humidity, and it can be stored for 8 to 10 years. And breeding collection or working collection includes the germplasm which are actively used in the breeding program, and is provided with the temperature which ranges in between 5% to 10%, and the relative humidity is provided with 8 to 10%. And then in the next step, gene pool is developed depending upon the cross compatibility between the species or the germplasm we have collected, and like a primary gene pool which will include species which are 100% compatible, cross compatible, and secondary gene pool which requires sometimes some technique for crosses, and tertiary gene pool always requires special techniques to cross, while quaternary gene pool will always contain all those species or accessions which have 0% affinity to cross with each other. And then finally, accessions are evaluated on the basis of scientist interest, our future needs, and some other parameters. However, these are specially evaluated on the basis of yield components, height, growth habits, time of flowering, resistance to genes of resistance, etc. And finally, after all type of evolution, Now we will get a pre-breeding population having genes of our interest, and we can use this population in our breeding programs to develop new cultivars. And here is the conceptual diagram showing the steps for all this procedure, and here are the references. And if any one of you have a question, kindly ask us via these emails.